We present the visual analytics dashboard for the integrated exploration and sense-making of cancer cohort radiogenomic and clinical information. Our motivation is prostate cancer, a very common disease in the biological male population. It's developed in 1 out of 6 persons, and still its exact cause is not known. Leading risk factors are investigated in 3 different datasets of radiomics, genomics, and clinical scores. Therefore, we enable the integrated analysis and exploration of these 3 datasets to gain new insights from the data. These data consist of imaging-derived radiomic features and gene mutation data, known as radiogenomics, and clinical scores. We allow cancer experts and biomedical data scientists to gain new insights from the high-dimensional and heterogeneous radiogenomic and clinical data by 4 different tasks. In the pre-processing, we prepare the data for the automated analysis and visualization. The cohort stratification divides the patient data into groups of similar radiogenomic and clinical profiles to identify new patterns in the data. The free exploration supports the understanding of these patterns and serves for knowledge discovery. In the hypothesis-based exploration, we allow users to assess and refine hypotheses on the data. Our framework consists of a main view with a scatter plot. Here, each of the points represents one of our patients. Users can interact with the patient through zooming, through processing any patient subset for the cohort stratification, or through comparing any active selection on the scatter plot with patients that are not currently selected. To understand the groupings in the scatter plot better, we present the heat map with the characteristics and differences of both clusters. Features with the highest impact on the clustering can be identified by the dark red color. A tooltip shows feature distributions of the clusters on demand, and the mouse click highlights the feature values on the scatter plot. The clusters view provides a detailed overview on the features differentiating these clusters. Users can switch the view to investigate characteristics of the green or the orange cluster instead, or filter any of these views on demand. The top genes view presents a ranked list of gene mutations expressed for most of the patients for any active patient selection on the scatter plot. The processing view, users can define any dataset to include or exclude from the analysis. The hypothesis view allows the assessment of statements. For example, we can identify that only one patient has both the MET12 and the PLEC gene mutation. We can also assess all patients that have at least one of these 2 gene mutations to identify that they have an isope grade of at least 2. We can see that the sphericity feature highly correlates with the clustering on the scatter plot. Advanced analysis options are available on demand. These allow users to investigate the impact of any change of the analysis methods on the resulting patterns and patients. We evaluate the data imputation methods through calculating the root mean square error and assess the cohort stratification through cluster similarity scores. We evaluate our framework also together with cancer experts by usage scenarios and by experts interacting with the application. The expert feedback confirms that our framework reveals interesting insights on the data. It helps them in exploring and understanding relations in their datasets.